All right, we're in chapter 5, section 4, and we're going to talk all about antibodies and how we use antibodies in biochemistry. So I know we talked about this earlier. You know, we briefly touched on antibodies, and we said, okay, we're going to tell later about how we use them, right? And so we know that antibodies are large multi-subunit proteins, and they bind to antigen molecules, right? So antibody binds antigen. And the really important thing is that you realize that antibodies have high specificity. So let's write it down here. Antibodies have high specificity and affinity. So that means that they bind only to certain things, and they bind very tightly to those things. So it makes them super useful in biochemistry, because we can identify specific proteins in really complex protein mixtures like cell extracts and tissue samples. But we can get really specific information even though we have this massive mix of proteins. So before we discuss how we use antibodies as a biochemical tool, we're going to talk about the relationship between antibody structure and antibody function. Okay, so we'll understand how antibodies work and then how we use them. So antibodies, if you've taken some biology, you know they are produced by B cells in our immune system, right? And each B cell makes only one single type of antibody. And B cells are producing antibodies that recognize only foreign particles. So any antibody that is made, or any B cell that makes an antibody that recognizes us, our own proteins, or self, those are killed, they're eliminated. Because if they're not, then what we end up with are autoimmune diseases, because we don't want our immune system to recognize our bodies. We only want them to recognize foreign things. So B cells can make two classes of our light chain, which are lambda and kappa. And I don't know why your book did this, but really it should be a I don't know if they tried to do that as an L, but it should be an I, right? So two classes of Ig light chains, so lambda and kappa, and then you have 5 classes of Ig heavy chains. And I'm going to remind you of what the light chain and heavy chains are in just a second. And these are all of these Greek letters, but typically in biology, you're going to hear them called, instead of mu, you're going to hear it as Ig M, and then alpha is Iga. Then we have delta, which is IgD, then epsilon, which is IgE, and then gamma, which is IgG. So you probably heard IgG before, right? But they're just different kinds of antibodies. So let's go back real quick. This is from your slides from 5.2, I believe. But this is the same slide you had before, right? So it just shows us the structure of the antibody. So remember that antibodies have light chains and heavy chains. They call them light chains and heavy chains. Why? Well, if we look at the blue, right, the blue is much longer, and therefore it's going to be heavier than, for example, the red chain. The red chain only has one constant region and one variable region. The heavy chain has 3 constant regions and 1 variable region. So that's why we call them heavy and light chains. So if you look at the light chains, what you're going to see is that we have 2 domains, right? This is our variable domain. So we call it V sub L, variable light. And then we have a constant chain. The constant chain is C sub L, so constant light. That's what the abbreviations are. If you look at the heavy chain has 1 variable region, so that's VH. Then it has 1, 2, 3 constants, so that's CH1, CH2, and CH3, right? So how does an antibody bind to an antigen? The way an antibody binds to an antigen is that the combination of the light and heavy chains variable region, so the combination of the variable and heavy chain, the combination of the light and heavy variable chains make up the antigen binding site. So this little yellow, is that a hexagon? Binds to those two variable regions. And so that's why antibodies, you can make all kinds of different ones, right? And they recognize very, very specific sequences because this variable region is so variable from antibody to antibody. Okay. So in the lab, we use protein-specific antibodies as tools, right? So these protein-specific antibodies are generated by immunizing animals with purified proteins under conditions that initiate an immune response. So when we do this, we want our antibodies to target a particular place, right? We want to have antibodies for our experiments that are very, very specific, that are only for the protein that we are really interested in studying. And so in order to do that, we need to look at those antibodies and know what they're recognizing. So when we look at an antibody, we know that the specific site where that antigen combines antibody is an epitope. So when we make these antibodies, you have to realize that one antigen, so here's an example of one antigen right here. There can be multiple sites on that antigen that can be recognized by an antibody, but each specific antibody only recognizes one epitope. But because antigens can have multiple epitopes, right? You can have multiple different antibody molecules that recognize the antigen. And this is, I think immunologically speaking, this is a good thing, right? You don't want there to be only one antibody that can recognize a virus. You want your body to be able to produce many antibodies that can all recognize the virus, but all in different places on the virus so that the virus can't escape your immune system. That's the physiological background behind why. So, and the other thing to kind of think about also is that epitopes are usually on the outside and they're polar and contain kind of within loop regions because they vary a lot. And so you can make antibodies to very specific things because they're so variable. All right, so we're going to get an animal and we are going to immunize it with our antigen, whatever that antigen is. Maybe it's something I purified, maybe it's something I produced in the lab, right? And I'm going to get it to make antibodies. Well, there are two different kinds of antibodies, right? So maybe you took that polypeptide that you were really interested in studying and you cloned it into a bacterial expression system, or you did solid phase peptide synthesis. Either way, you're going to generate antibodies with it, but you can generate two kinds of antibodies. And we're going to talk about how they're different and what we use them for. So the two kinds are polyclonal antibodies and monoclonal antibodies. In polyclonal, we have all different antibodies that all recognize one antigen. So in polyclonal, you have many antibodies, and that's how I abbreviate antibodies. You'll see that a bunch of different places. So you have many antibodies that recognize one antigen. And your antigen is your protein of interest in this case, right? But each antibody is only going to recognize one epitope. So if you have many antibodies that recognize that antigen, you're probably going to be recognizing multiple epitopes. So polyclonal recognizes multiple epitopes. This is the kind of key there. In comparison, when we look at monoclonal antibodies, right, these are only going to recognize one epitope. So this is one epitope on one protein. So this is one antibody, one epitope, right? This one is many epitopes. Okay, so that's where the poly and the mono come from. Poly meaning we're going to recognize many epitopes, mono meaning we're only going to recognize one. But the procedure to generate these two is different. And so we're gonna talk about generation of polyclonal first, and then we'll talk about how monoclonal is different. So we get our animal, and it can be a rabbit, a chicken, goat. Those are, I think, the most common ones. But so you immunize the animal with your protein antigen, and you do it a couple of times over like a 6 to 8 week period. And this is exactly what happens when you get a vaccine, right? The animal's immune system is going to recognize the peptide as foreign, and then it's going to amplify B cell populations, right? Ramp up B cell production. Then those B cells are going to express antibodies that have high affinity for the antigen, right? Now, that animal is carrying that stuff around in the blood. So what you want to do is you want to isolate the blood serum. So you take blood samples out, you remove the red blood cells. What you're left with is that blood serum that has high amounts of both antigen specific, and unfortunately you also have non-specific antigens, right? It's not the only antigen that the animal's producing. Hopefully it's the one that it's producing the most of, but it's not the only one. So then, because this is a mix of antigen specific and antigen non-specific, PE antibodies, right? So you need to purify it. You only want the antibodies that are going to recognize your protein of interest. So we purify them using affinity chromatography. So we attach our peptide to beads, right? So what you have here, you have a bead, and you have, this is your protein. What is this? YOPI, your protein of interest, right? And you put your serum over this column, right? And you let the flow through go through. So anything that didn't bind to YOPI will get eluted out. Then you're going to wash it, because you want to make sure that you've only got super specific antibodies in there. So you wash it, you let all those non-specific antibodies come out. So you don't want to keep those, right? Now you can actually elute out specific antibodies. So these are the ones that actually bound to your protein of interest, right? So these are the ones we want to use. So this is polyclonal. We know that this antibody is binding to our protein of interest, but is it recognizing the same epitope? We don't know. Most likely no. Most likely you have a mixture, and this is why we call it polyclonal, right? Because all we know is it's that these antibodies are binding to your protein of interest. We don't really know where, we don't know how many different ones, but that's why we call it polyclonal, because it could be multiple. So now let's look how it is different in monoclonal. So in monoclonal, our initial steps very similar. We're still going to immunize our animal, right? I don't know why they did. Well, I guess rats are not that common, but it doesn't matter. Anyway, so the generation of your monoclonal is similar. We immunize the animal. You know, we give them boosters, and once we get them producing B cells and producing antibodies just like before, then we want to start to harvest things. But instead of harvesting blood, we're going to harvest the antibody expressing B cells. We don't want the antibodies, we want the cells expressing the antibodies. So we are going to specifically isolate B cells. And usually it comes from the animal spleen tissue. And we fuse these isolated B cells with tumor cells. Now, the great thing about tumor cells is that they're long living, right? You can grow them in culture forever and ever. There are some that have been growing in culture since, like, I don't know, 1940 something. So they've been going for a while. They're called immortalized, right? And so now we have this combination cell type, and we call this a hybridoma. So a hybridoma. And a hybridoma is hybrid, right, between the B cell and the tumor cell. So we keep the property of producing B cells, but they can stay in culture for a really long time, right? So now we can culture these hybridoma cells and screen them for antibody production, right? Because we only want hybridoma cells that are expressing the antibody we want. So basically what we do, and I don't like how they did this, right? You end up with this tube and you've got this mixture of all these hybridoma cells. And so what you do is you plate them out such that, and you could do it in a well, or you could do it however you want, right? But this is a well plate, so that each well only gets one type of cell, right? And then you let it grow. And so you end up with populations that are clonal. So clonal populations come from one cell, right? So in each well of this, you only started out with one cell, so you know all the cells are exact duplicates of that same cell. So now you have different cultures. So each hybridoma cell is going to secrete a single antibody species. And that single antibody species is going to recognize, guess what? A single epitope on the antigen. And so now you can establish a clonal hybridoma cell line. So you can propagate this one cell line that you really like. But what you want to do is you want to make sure to test for specific binding activity. So you want to make sure, okay, I'm going to take a sample out of this one, right? I'm going to increase the number by just growing them in culture. And now I'm going to test it. And I'm going to check to see, I'm going to look at the supernatant, right? Because they're going to produce those antibodies and it's going to go out into the media of the cell. And I can test those antibodies and see, are they binding to YOPI? Are they identifying the protein that I want them to? Are they binding on the correct binding site, right? So this process is expensive. This process is time consuming. And there is no guarantee that you will find a monoclonal antibody. But if you do, if it is successful, you not only have an unlimited supply of the antibody, because these cells can grow forever in culture, but your biochemical results from using monoclonal antibodies are going to be higher quality and more specific. So it generates better data. So they both have drawbacks and perks, just like everything else. So now, a common application of antibodies in the lab is called Western blotting. So in Western blotting, you're going to have your protein. It's usually not just your protein. It's usually you have a cell lysate, a mixture of cells, right? And so you run a PAGE gel, right? Gel, this is a PAGE gel. And it could be a 1D, it could be a 2D, it could be native, it could be denaturing, doesn't matter. But you run a gel. So you're going to separate them based on either just size or size and charge, doesn't matter. But you have to get that initial first separation. And then you're going to use a combination of two different antibodies. And we call these antibodies primary. Primary antibodies recognize YOPI, right? Your protein of interest. The secondary antibody only detects other antibodies. And we're going to talk about how that works. I'm going to walk you through the whole procedure. So here's kind of our schematic of how things work. So the first step is we're going to run our gel, right? We're going to separate our proteins based on size or charge. So here's our gel. We've run our gel. And we have kind of separation of our proteins, right? And we also need to make sure that we have a molecular marker on the side so that we have known proteins of known size so that we can look at, okay, it was this size or that size, right? So now we transfer our proteins to what's called a nitrous cellulose filter membrane. So here's our filter membrane right here. And you use, you want to use this membrane because the proteins are going to bind to that membrane with super high affinity, right? And so the really cool thing is wherever, if you lay this gel and lay this membrane to where they're touching, what's going to happen is that these proteins get transferred from the gel onto the membrane. But look at this, they stay in their locations, right? So the proteins retain their original positions on the membrane relative to the original gel. So they haven't moved, they just transferred from the gel to the membrane. Now you take that membrane and you actually add, and your book doesn't say it right there, but right here, you want to add a blocking solution, right? Because this blocking solution prevents non-specific antibody binding, right? Some antibodies will kind of randomly bind to stuff, and so you want to decrease that. So you treat it with a blocking solution, then you incubate your membrane with what's called your primary antibody. Your primary antibody recognizes Yopi, right? You wash away to make sure anything that was unbound, you get rid of, right? Then what you do is you incubate the membrane with an enzyme-linked secondary antibody. This secondary antibody is called the detection antibody. And this detection antibody recognizes your primary antibody. Recognizes primary antibody. And so if, let's say, for example, your primary is a mouse monoclonal antibody, right? Our primary here was mouse. Then your secondary here will be from a different organism, so a rabbit, and it'll be a rabbit polyclonal. So we call this rabbit anti-mouse. So if your primary is, what did I say? Primary is mouse, right? Then your secondary would be, it has to be from another organism, because if you took it from a mouse isn't going to recognize mouse antibodies as foreign. So you need a rabbit anti-mouse. Right? But what's really interesting about our secondary antibody is that it's not just a secondary antibody. That secondary antibody is linked to an enzyme. And this enzyme, in the presence of a certain substrate, will give off a color, right? Or some kind of a detectable product. And so if you have that secondary antibody that's covalently linked to the enzyme, you can use a colorimetric, a fluorometric assay where it's going to light up, right? Our protein of interest is now going to light up. So here's our protein that's covalently linked to our enzyme. And when we expose it to whatever method you so choose, because there are multiple out there, what you're going to see is only those specific, and I don't like this, I don't like this last one, because you should only light up your protein of interest. If this is the only place on the entire gel where our antibodies recognized everything, this is the only place that should be lit up on our last gel. So this is the only product you should see. You're not going to see actually any of this, right? None of these lit up, so none of these are going to be visible. So at the end, you're only going to see things that were bound, number one, by the primary antibody, and then visualized with the secondary antibody, right? So by comparing the position of where our secondary antibody on the membrane is, right, where did it light up, we can compare that to our original gel, and we can determine which protein samples contained our target protein. So if I were to do this, I would look like in this lane, and this corresponds to, I think, this lane, right? And I could say, okay, I know in this lane I had untreated cells. In this lane I treated with drug X. This lane I treated with drug Y. Oh, when I treated with drug Y, my protein of interest was expressed, whatever it is. So that you can tell what conditions resulted in recognition of your protein, right? So you can get a lot of information out of a Western blotting scheme. So let me show you another application of Western blotting. Let's just say that you have a protein coding sequence and you don't have an antibody for it. It's kind of sad, right? You can't run your experiment. But what you can do is you can put your protein of interest into a cloned cloning scheme, right? You can make it, and you can actually put a tag on it. You can add any kind of tag that's going to be easy to recognize by an antibody. So 2 examples here are called a flag tag and a MIC tag. And so when you look at these, you take your normal protein sequence and you insert this little piece of DNA, right? And this DNA codes for a certain, in this case, this is aspartate rich protein sequence. Now this aspartate rich protein sequence is recognized by a primary antibody that is specific to the flag tag, right? So you want to monitor, you can clone this into any cell. You can clone this into a human cell that you have growing in tissue culture. And you want to monitor under X conditions what happens to your protein. But if you don't have a primary antibody for it, you can't do it. So you can generate a recombinant protein that has this tag and you can monitor it. You can see what happens. Does the expression go up or down when I do X, Y, or Z, right? And the antibodies for these flag tags or MIC tags are commercially available. And so you can use them in your studies, but you have to be very careful. You have to understand that even adding a small sequence like this aspartate rich sequence or this glutamate rich sequence, either one can actually alter our protein function. And so a lot of times what we do is we test and see, is it better to put it on the N terminus? Is it better to put it on the C terminus? Where would it alter protein function the least? And so that's something you kind of have to keep in mind because you want this tag to kind of hang out on the outside, otherwise it won't be recognized by the antibody. So usually these tags tend to be polar and they stay, you want them to stay on the surface of the protein. So if they're polar, they won't get folded in with all those other non-polar residues. So usually if they're polar, it prevents them from altering the structure too much, but it is something to kind of keep in mind. All right, so that's one example. Another example is using it in immunofluorescence. So this is another antibody-based technique, and we use this to identify proteins in cells that have been chemically treated. So they're not living anymore, but we're going to preserve the cell architecture. And so we use a primary antibody on cells that are already cross-linked to a microscope slide. So the cells have been, they're dead, they're preserved, they're stuck to the slide, and then we put our primary antibody on it. Then we wash the primary away and we put a secondary on top, and the secondary contains some kind of a fluorescent group. So let me show you what that looks like. So here's an example where we have here, so the, I'm trying to remember, this one's the red. Okay, so the red here, as I draw it in blue, but the red is a primary antibody for a cytoskeletal protein called cytokeratin, K-E-R-A-T, keratin 8. And this is human cells. And so you have this primary antibody to the cytokeratin 8. You wash it, right? Then you put on a secondary antibody. The secondary antibody has a covalently linked fluorescent group. And there are 2 different fluorescent groups that you could use, Alexafluor 488 or Alexafluor 594. And those are just the wavelengths that you're exciting these 2 molecules at. And so depending on which one you use, it's going to emit either red light or green light after laser excitation. In this case, they used the red one, so they used the 594. And you can actually use this in combination with other techniques. So what's really cool about this one is that they used this in combination with another fluorescent technique. And so they used a protein called phalloidin, which specifically binds to actin. You all know actin is in the cytoskeleton. And so this phalloidin is actually tagged, and it's got a covalent, V-A-L-E, valent tag attached to it, right? And that tag is a fluorescent group. And so this tag is green in this case. So they wanted to look at, in this picture, where is this cytokeratin 8 in relation to the actin cytoskeleton? And so you tag both at once using different tagging techniques, and you can overlap the two images, and you can see, do they co-localize? Are they in different places? Are they in the same places? So you can get a lot of information that way. So this is a really cool and fun thing to do, I think. I love it. All right, so here's another one, ELISA. This is another way we can use antibodies. We know that antibodies are super, super specific, right? So they're really ideal for identifying proteins that might be present only at very, very low levels. So this is good for low levels of protein, right? Let's say YOPI is at very, very low levels. We can use ELISA to detect the protein, even though there isn't very much of it there. So how does it work? Well, you're going to use two different specific monoclonal antibodies to detect our protein of interest. And what's cool about this is that you can do this in a 96-well plate. So you can have lots of samples at one time. And we haven't had that ability up until now. And so this particular method that I'm going to tell you about is called a sandwich ELISA. A sandwich ELISA. Yum. Anybody hungry? All right. So you have a primary antibody, and it's called the capture antibody. So that's this one right here, our capture antibody. And we attach it to the bottom of the 96-well microtiter plate. And so let me erase this. So that primary antibody is going to capture our antigen, or our YOPI, our protein of interest, right? Because we're going to put cell lysate, we're going to put whatever condition you put your cells in, but you add it to the well and you incubate it for a couple of hours to help make sure that antibody's binding properly, right? And so you have your antigen stuck to your primary antibody on the bottom. Now you're going to wash it. You're going to make sure to get anything that's not bound to that primary antibody out well. Then you're going to add a secondary monoclonal antibody. So this is a secondary, it's not a secondary, it is a primary monoclonal antibody. Now, these are both, the capture antibody and the detection antibody are both primary monoclonal. But the thing is that these two have to recognize different epitopes. Because if they recognize the same epitope, then the detection antibody would have nowhere to bind because the primary antibody already bound it. So you have to have different epitopes, right? So your detection antibody and your capture antibody have different epitopes. That's super important. So once you have that detection antibody attached to your YOPI, your protein of interest, right? Then you add, here's your secondary in red. So this one right here is your secondary antibody. So we have our secondary antibody, and our secondary antibody is the one that's hooked to that enzyme. And a lot of times that enzyme is called horseradish peroxidase. And so when it has a particular substrate present, the enzyme will convert it to a product that is fluorescent, right? And so using this method, right, you can do lots and lots and lots of experiments like on one dish, right? You can try different conditions. You can do all sorts of different things and get a lot of data and a lot of samples all at once. And you get like a positive or negative inside of the well. Either the antigen was present and all of the other stuff attached to it afterwards, or the antigen was absent and all you're left with is on the plate this primary monoclonal capture antibody, and that's it. So it's kind of a plus or minus sort of experiment. All right, I think the last one we're going to talk about is immunoprecipitation. So this is cool. This is really, really cool. You can use immunoprecipitation to purify a protein, or really to purify things that are associated with your protein of interest. So it's actually a combination of two biochemical assays. So what we do is we use high affinity antibodies to isolate our protein of interest, right? So we have a monoclonal antibody, right? And we covalently attach it to a bead. And so what they don't really show you all the things that happen in it, but you have this monoclonal antibody, and I'm going to draw it for you, and we're going to attach it to a bead, right? So this is monoclonal, right? And we attach it to our bead here. And so you incubate this primary antibody with our cell extract. So we add our cell extract, right? And inside our cell extract, we have this typically not, Yopi is usually not all by itself. Yopi is usually in association with a bunch of other things. And so we're assuming that Yopi is number one in this complex, right? Number one is equal to your protein of interest, right? And so when Yopi binds to our antibody that's stuck to the bead, well, all the other stuff that's hooked to it stays with it. So now that we've incubated it, the bead's not attached to anything else, so it's just kind of floating around. So what we can do is we can spin it. We can get it to settle at the bottom of our centrifuge tube, right? And we can kind of wash away all the junk that we don't want. So now we have basically just our primary antibody, Yopi, and anything that's associated with Yopi. Now we can take that and we can run just what's on that antibody on an SDS page gel. So remember, we do need denaturing conditions, because now we want Yopi to let go of protein 2, protein 4, protein 3, whatever those other things are, so that they'll separate on an SDS page gel. Now that we've got that separation, well, that's great. So what we can do is say, all right, I know Yopi's number one, Yopi's number one, but I don't know what all this other stuff is. So what I'm going to do is I'm going to cut my gel out. I'm going to cut that little square out. I'm going to take it out, and I'm going to trypsin digest the protein that was in there. And once I trypsin or chemotrypsin, whatever it is, right, I can then analyze it with mass spec, and I can determine what protein was actually there, right? I can compare the sequence, and then I can do it for my other proteins, right, who are in here. Then I'm going to repeat it with this one. And then I'm going to repeat it with my pink one. And I'll be able to identify the proteins that associate with Yopi. That's really, really important information. And so a lot of times people call this co-immunoprecipitation, because what you're doing is not just immunoprecipitating your protein of interest, you're immunoprecipitating your protein of interest in complex with all kinds of other things. And so you can get lots of information about the interaction partners for your protein of interest. And so it gives you a whole lot of information. All right, and I think that is it. Oh yes, that is it. All right. Have a great day.